Today I'll be talking about estrogen as a contributing factor to the development of lipedema. I have nothing to disclose. So as most of you now know that lipedema is now recognized as a loose connective tissue disorder. It occurs almost exclusively in women. It begins at the time of hormonal changes, puberty, pregnancy, and menopause. It's characterized by painful bilateral and symmetrical expansion of the fat in the lower extremities. Hands and feet are spared, and fibrosis and lipedema are common complications of lipedema. And here is an example of lipedema stages, which all of you are familiar with. I just want to note that changes in the skin texture as we move from stage 1 to stage 2 to stage 3. But what actually do we know about the lipedema fat tissue itself? And here I'm showing that the just comparison in the fat between normal fat and lipedema fat. And then you can notice in the lipedema fat, we have the fat is firm, uneven in texture, and then you can see and feel the nodules below the skin. And when we did an in-depth analysis of that tissue back in 2019 with Dr. Karen Herbst, we have shown that lipedema tissue is basically have larger hypertrophic adipocytes, which is larger fat cells as compared to non-lipedema patients. And then we also have shown that these lipedema patients have higher or increased number of blood vessels, both in the epidermis and dermis, in both obese and non-obese lipedema as compared to non-lipedema patients. We have also shown that these patients have more dilated blood vessels. They have angiogenesis by increased endothelial cells and SMA. We have shown that they have high fibrosis. We have interstitium increase, which increase in the spaces between the fat cells. We have shown that we have high incidence of macrophages and mast cells in these samples. This is at the tissue level. When we went and isolated cells from these patients, and here we isolated stem cells, and these are the cells that have the ability to differentiate into fat cells, we have shown that cells isolated from lipedema have the potential to have higher adipogenic potential. That means they have higher ability to be induced into fat cells as compared to non-lipedema cells. And when they isolated these cells from the thigh, we have shown that lipedema ISC express higher leptin as compared to non-lipedema ISCs, and leptin, you know, is a hormone that's secreted from fat cells to regulate the body weight. We also have shown that when these cells are isolated from the abdomen, they express higher PPR gamma in lipedema as compared to non-lipedema ISCs, and PPR gamma is the main adipogenic marker that regulates fat cells. So this is what you know, but since our aim is to find a treatment for lipedema, we need to first know what triggers basically both increase in fat, inflammation, and fibrosis in lipedema adipose tissue. One hypothesis that we've been working on is that hormone, and in particular estrogen, might impact the development of lipedema. And here is based on the following facts. First, that we know lipedema manifests during periods of hormonal changes, and then estrogen itself increases the expansion of fat tissue. So when we have normal levels of estrogen, we see increase in number of the cells, but we don't see inflammation or fibrosis. However, when we have low level of estrogen, we can see increase in cell size, we can see adipogenesis, inflammation, and fibrosis, and this is which we see similar in lipedema patients. Second thing is that estrogen itself exerts its effect on the estrogen receptors that are located inside the cells and on the cell membrane. And estrogen receptors have been shown to be differentially expressed between the upper and lower body fat, again emphasizing on the role of estrogen played in lipedema and played on female body fat distribution. Studies have also shown that estrogen might regulate the adipose stem cell proliferation and their ability to differentiate into fat cells. So thus, our work plan is to determine the expression of estrogen receptors and its metabolizing enzymes in lipedema, both in fat tissues, we have them isolated from the thigh, in pre- and postmenopausal women, as well as in the cells, ASCs and adipocytes, to look at their gene expression, protein expression, and then we want to study the specific effect of estrogen on these cells, and this is when we treated the cells with estrogen and see if they have the ability to basically differentiate into fat cells. So this is just, I know it's a busy slide, I just want to show two things, that estrogen, it has mainly three forms. It's E1, E2, and E3. E2 is the major estrogen, basically circulating and biologically active form of estrogen. The two main receptors that I'm focusing on for this talk is estrogen receptor alpha and estrogen receptor beta, and all these like metabolizing enzyme, their role is either to activate estrogen or deactivate estrogen during premenopausal or postmenopausal cycles. So to start with, we had tissues, thigh tissues, collected from premenopausal women and postmenopausal women from basically controls, lipedema stage 1, lipedema stage 2, and lipedema stage 3. Unfortunately, we don't have any L3 in premenopausal or any L1 in postmenopausal. But what our study have shown is that in lipedema premenopausal stage 1, we have an increase in adiponectin, the main fat gene expressed by the fat cells, increase in ER alpha and ER beta as compared to non-lipedema cells or lipedema stage 2. However, there is no major changes in postmenopausal women. When we looked at one of the metabolizing enzyme that basically generates E2, we have seen that's increased again in premenopausal women lipedema stage 1 as compared to non-lipedema. However, there was no major change in postmenopausal women. Interesting, when we looked at more enzymes, and here I'm looking at LIPE, which basically generates E2, it is significantly increased in lipedema stage 1 premenopausal women, and in SULT-E1, which basically decreases in postmenopausal, lipedema stage 3 women. And this one enzyme is very interesting because its decrease is involved in increased inflammation and adipogenesis. And this is what's happening at the tissue level. But when we went on and look at these cells isolated from patients and see what is happening in vitro or in their body, we took these cells first and isolated them and put them in a hormone-depleted media. So this media, so basically cells are cultured in their normal media that favors their growth. What we did is we cultured them in a hormone-depleted media. So we depleted the media from any estrogen or androgen, and we looked at their ability to grow and proliferate in culture. And here what we have seen is the number of cells decrease, which is basically quantified in here. So if you look at the red line and green line, you can see the major decrease in proliferation both in healthy and in lipedema cells. But what's interesting is when we treated these cells with E2 to see if they recover, only healthy cells respond to the treatment, so they grow again when we add E2, but lipedema cells did not respond. So that means lipedema cells did not sensitize for estrogen treatment. So again, when we looked at we went further and looked at the estrogen receptors in these cells, both in healthy and lipedema. And what I'm showing in here is just that estrogen receptors both increase their expression when both of these cells are treated in hormone-depleted media. So the media is free of estrogen, so the estrogen receptors now are free. What's interesting is when we treated them with E2, there was no effect on estrogen receptors. This at transcription level, and we look at the same estrogen receptors at the protein level, and we get the same data. Estrogen receptors are increased in hormone-depleted media. But what's interesting is when we treated with E2, we've seen a decrease in ER beta expression in lipedema. So that means there is a potential treatment in here for lipedema cells. We looked at the other, again, these enzymes that basically activate estrogen. We have seen that all of them are increased in hormone-depleted media. But whatever is interesting in here is that in lipedema cells that are treated with E2, we have increase in hydroxy steroid 17B7, and this is important because this enzyme in particular is increased in breast cancer patients too. When we looked at more enzymes, in here, again, they are increased in hormone-depleted media, but what's interesting in here is when we treated with E2, we have seen that aromatase, which basically converts androgen to estrogen, is only increased in healthy cells, but not in lipedema patients. So this is what's happening at the stem cell level. And then we wanted to see, is this the same thing happening on the fat in the adipocyte cells? So we treated these fat cells with E2, and we look at the expression of adipogenic marker and the estrogen receptors, and we have seen that only in lipedema adipocytes we have increase in PPR gamma 2 when treated with E2, and this is basically the main adipogenic marker. So estrogen induces adipogenesis in lipedema. And then when we looked at the estrogen receptors, we have seen that they are only increased in healthy cells and not in lipedema cells. So for the enzymes, we have seen only B1 increase in healthy treated with E2, but there is there is nothing increasing in lipedema cells, so they are not activating estrogen anymore. So basically, to sum up, I know it's a busy slide, I'm just saying that when these cells are, when the culture in hormone-depleted media, we have seen a decrease in the proliferation, so decrease in their growth. We know that estrogen failed to induce proliferation of lipedema ASCs in hormone-depleted media, but compared to the healthy ASCs, we have seen that estrogen upregulated aromatase in healthy ASCs, but not in lipedema, and estrogen increased the expression of B7 in lipedema ASCs. Also, it's increased its adipogenic marker, which is PPR gamma 2. So again, since our aim is to find potential treatment for lipedema, I think targeting estrogen metabolizing enzyme could be a potential treatment. However, we don't have to forget that lipedema, again, is a complex disease. So if targeting only estrogen receptor might not be the ideal treatment for lipedema patients, so if we find a combinatorial treatment that basically include both inhibitors of angiogenesis, inhibitors of inflammation, and estrogen might be ideal for lipedema treatment. Finally, I would like to thank my lab, my collaborators, and the Lipedema Foundation for funding this project. Thank you.
We have time for a couple questions. One of the questions is, what about estrogen mimicking foods like soy? Can this affect lipedema progression too? Or is there any research that supports this?
There is no research yet for this, but I would expect yes.
Okay, fantastic. And another question that came up too is,
you can hear me?
Just step away. Okay. Yeah, a little too close with both the microphones. Since fat and the disease lipedema are estrogen producers, do we know which came first?
That's a very good question. No, we don't have an idea, but hopefully we will try to figure it out. Thank you.
Thank you so much.